my name is Joanna Yeh, and I'm a researcher at the Mass General Hospital and Harvard Medical School. And I think in the last 3 days, you have heard about all these very different flavors of Cas platforms and the amazing things they can do in human cells. So today, I'm here to tell you a little bit about how we apply CRISPR technology into the zebrafish model. But before we get into that, I think it will be important to know why we are interested in this model organism and what are the advantages of it, so that what do we want to do with it. And first, I guess, may I know if anyone here are using zebrafish in their research? Okay, so I hope that no matter you are currently using zebrafish or not, you will all maybe find something interesting in this lecture. And let's see. So in the last 10, 15 years, zebrafish has emerged as a very popular vertebrate model organism, as evidenced by the numbers of publications during each of the 10-year period from 1990. Let me find it. And this is partly because that zebrafish is actually quite a suitable model for the studies of human diseases as well as for drug discovery. After all, zebrafish exhibit important molecular similarities to humans, and about 70% of human genes and as well as 82% of the disease-associated genes in human disease-associated genes have zebrafish orthologs. And zebrafish also utilize similar physiological systems as we do. And the mutations in the genes that associate to human diseases also induce disease-related phenotypes in zebrafish models. And most importantly, zebrafish sit in this sweet spot that it has the biological complexity that some of the invertebrate model organisms don't have, but it also still enables high throughput experimentations. So if we dive a little bit more into the zebrafish genome, it contains about 1.5 billion base pairs, which is about a little less than half of the size of the human genome. However, it has approximately 26,000 annotated protein encoding genes, which is more than the about 20,000 protein encoding genes in human genome. And that is because that zebrafish ancestors have gone through a partial genome duplications. So about 3,000 human genes have either 2 or more zebrafish orthologs. But also that zebrafish seems to have a very high number of fish-specific proteins. However, if you just look at the the genes that are orthologous to human genes, the zebrafish has orthologous genes that's higher than the chicken model, but lower than the mouse model. And then if we look a little bit more closely about the advantages of the zebrafish model. So we've mentioned the molecular and physiological similarities between zebrafish model and human. In addition, zebrafish are fecund. One pair of fish can lay about 100 to 500 zebrafish embryos in a morning. For example, this conical tube contains about 10,000 zebrafish embryos that we can collect in one morning. And zebrafish also have very rapid ex utero embryonic development. And because their embryos are transparent, they enable in vivo real-time imaging during their development. So once the zebrafish eggs hit the water and become fertilized, just within the first 3 hours, these embryos gone through 10 cell divisions. So their development is really fast. And then within just 1 day, 24 hours, zebrafish embryos already have eyes, ears, brain, and then you can already see beating heart and their circulating blood in the developing embryos without any dissection. And you can also do the lineage tracing using fluorescent reporter lines, and then do, again, real-time imaging while they develop. This capacity, zebrafish embryos stay transparent for about up to 5 days. And only 3 days after they are born, they become free swimming. And 5 days after, they become, all their major organs are developed, and they can already start to seek food. They can respond to external stimuli. So you can do all sorts of behavioral assays already at 5 days old. In about 21 days, they also already exhibit social interaction behavior. You can analyze their social interactions. However, unfortunately, the generation time of zebrafish is actually not shorter than the fish model. Fish normally, the adult fish normally don't breed until they are 3 months old, so their generation time is still similar. But because of these nice features, they are excellent models for embryonic and larval study. So in addition to these great features, because zebrafish embryos are small and they live in the water, so they also enabled chemical high throughput screening. So exactly how small they are. This image shows you 2 one-day-old zebrafish embryos arrayed in a well of a 384 well plate. So in general, chemical screening in zebrafish are done using 96 well plate, and you can array about 3 to 10 embryos per well. And then the compounds from a chemical library can then be added into the wells. And then you can observe the compound. You can identify the compounds that can induce specific phenotypes, or if you use the mutant fish that has already a phenotype, you can identify potential chemical suppressors. So the advantage of a whole organism phenotype-based drug discovery approach compared to, for example, the traditional target-based approach is that you can design the chemical screen even without a prior clear understanding of a disease mechanism. And that also that only a whole organism can be used to model more complex diseases that it cannot be modeled in a petri dish. Such as cardiovascular phenotypes or, like we say, social behavioral phenotype. And then sometimes when even we know the cause of the disease, but if the pathway has no known a clear druggable target, this will be also phenotype-based approach will also be the way to identify potential therapeutic targets. And because this screening is done in vivo, so potentially the effect of the chemicals, both their in vivo efficacy and toxicity, can be measured simultaneously. So using this approach, I'm just listing only a few examples here. Zebrafish screens have already identified some compounds that can either specifically inhibit BMP signaling pathway or suppress self-renewal of leukemia stem cells. It has identified compounds promote hematopoietic stem cell expansion, reverse Duchenne muscular dystrophy phenotypes or suppress doxorubicin-induced cardiomyopathy. So since zebrafish is such a useful model for disease modeling and chemical genetic approach, having the ability to targetedly manipulate zebrafish genome to create zebrafish models of human disease will allow us to study disease pathways, but also to identify chemical suppressors. And hopefully both of these approaches will may facilitate the development of therapeutics. However, like most of the other model organisms, the targeted gene editing had not been possible until the birth of ZFNs and talons, and then it only become like really possible for every single lab to make their most favorite mutants after the development of the CRISPR technology. So in the past, as Dana mentioned, in the past 10 years, I have been in collaboration with longtime collaborator Keith Zhang, who is also in Mass General, and Randall Peterson, who just recently moved from Mass General to University of Utah, on developing genome editing technologies in zebrafish. And previously, we have worked on ZFNs and talons, and more recently on the CRISPR platform. So in 2013, the first wave of CRISPR, the first wave of papers as reported that CRISPR-Cas9 can function efficiently in human cells. And we also reported that it can be used to efficiently mutagenize zebrafish genome. So in those early studies, we show that CRISPR can be introduced into zebrafish embryos by co-injection of in vitro transcribed guide RNAs and in vitro transcribed Cas9 mRNAs, and co-inject into one-cell stage zebrafish embryos. And we also, in the early studies, we use the bacterial SPCas9 sequences. And later on, we also use mammalian codon-optimized Cas9 sequences and found that it's slightly better. So in that study, we found that CRISPR-Cas9 can efficiently mutagenize 9 out of 11 genomic loci we tested. And we also found that it successfully mutated 2 of the sites on which previously we failed to target with talons. And since we know that the talons can be sensitive to DNA methylation, so we speculated that those sites might be methylated, and we verified that is the case by sulfide sequencing. So these results suggest that the CRISPR-Cas9 can target methylated DNA. And as expected, we also found that CRISPR-induced mutations can be efficiently transmitted through germline. And in many cases, we found that all of the fish that we screened, the injected fish we screened, are actually founders, and their germline transmission efficiencies can sometimes reach 100%. And importantly, we can also identify multiple alleles from one founder, and different founders may also carry different alleles, as here shown by the mutation sequences in terms of their insertions and deletions. So some of the guide RNAs are so effective that we can see complete loss of the wild-type allele in just the CRISPR-injected fish alone. And these results are from the fluorescent PCR fragment length analysis. We use this assay a lot. And so as you can see, since you know that CRISPR-induced mutations are mostly either insertions or deletions, so they will result in small differences in their fragment length. And so when you conduct a fluorescent PCR and run them on a DNA analyzer, you see the wild-type fish has one PCR product. However, in two CRISPR-injected fish, you can see a variety of mutant alleles occurred and the complete loss of the wild-type allele. And when the guide RNAs are very efficient, you can also directly see their phenotype in the injected embryos. In this case, we target tyrosinase, which is important for pigment formation. So you can see that CRISPR-injected fish have very almost no visible pigments compared to the uninjected fish. So, which means that this, because of its efficiency, it is possible to actually observe or assay for gene phenotypes in the injected fish. So we also use the fluorescent PCR fragment length analysis for identify founders and also for genotyping. So you can see here that using this analysis, you can see, you can identify the wild-type fish, the heterozygous fish, and the homozygous fish. And not only that, you can also determine their insertion or deletion size, so you can identify different mutant alleles. So since we use in vitro transcribed guide RNA using T7 promoter, so T7 promoter transcribed RNA all start with 5' GG dinucleotide. So that sort of limits the targeting range. So we also tested whether the 5' GG mismatch can be tolerated. And in this case, we study using the GG 5' mismatch, and then in addition to 18 nucleotide of the target sequence or 20 nucleotide of the target sequence. And first of all, we found that 5' two base pair mismatches can be tolerated because you see very high activities from all of those guide RNAs here. However, we also noticed that it seems that longer guide RNAs are longer than 20 nucleotides. So in this case, the spacer region in total is about 20 nucleotides compared to 22 nucleotides of the spacer regions. So longer spacer regions longer than 20 nucleotides seems to have reduced the activity. So from this, we would suggest that if you, in some cases, you have to have the GG or 1 or 2 mismatches in the 5' ends, you should still keep your spacer regions limited to 20 nucleotides. And then we also look at roughly about 130 guide RNAs and trying to determine whether there are some factors that affect guide RNA efficiency. So in these graphs, the blue bar shows that the mutation for the specific guide RNAs, the mutation efficiency is under 5%. And the red bar shows the guide RNA that can induce more than about 5% mutation efficiencies. And so in this, the first comparison, we look at numbers of GC nucleotides, because I think there's also some other studies suggesting that the GC percentage of GC nucleotides are important for guide RNA efficiency. And we show that there's no significant, so here is the first group is GC nucleotides, about 10 to 16 GC nucleotides in that 20 base pair spacer region, or 7 to 9. And we found that there's no big differences in their success rate. But however, if you look at the range of mutations that you can get with these guide RNAs, it does suggest that the guide RNAs with lower GC seems to have a little lower target mutation efficiency. Then we look at the number of 5' mismatches. And similar to our first initial study, so this group has no complete match, there's no mismatch. And this part contains either 1 or 2 mismatches. We found that with 1 or 2 mismatches, there's actually no negative effect on their success rate. And there's also no negative effects on their, no significant negative effect on their mutation efficiency. So I think you might also be aware of this publications that study was conducted by the John Lab showing that the CRISPR-Cas specificity or their off-target efficiency can be reduced by the truncated guide RNA. So using guide RNA that's shorter in spacer length, down to like 17 nucleotides, can sometimes reduce their off-target efficiency or making the guide RNAs more specific. The reason behind this is that when you have shorter spacer regions, then it may be less likely to tolerate of mismatches. So we also test that in the fish and found that if you reduce the guide RNA spacer length to 18 nucleotides, you do suffer a little bit on the success rate and as well as the mutation efficiency. However, 18 nucleotide of spacer regions, you still have sort of reasonable mutation efficiency. So from there, I'd like to tell you a little bit about our current platform of generating guide RNAs for using zebrafish. This is a more high throughput platform, and we found it to be quite efficient. First of all, we switched from T7 promoter to SP6 promoter only because that SP6 promoter can initiate from transcription from either GG or GA nucleotide, so that increased your targeting range a little bit, even though you can still allow mismatches. And then we use this cloning-free methods to in vitro to make the DNA templates for in vitro transcription, which would really enable us to make large-scale guide RNA construction. And then, in general, now we designed 4 guide RNAs per gene, and we co-inject them to ensure very high success rate and mutation efficiency. And I think this is actually quite important, quite an improvement in the efficiencies. We now start to use Cas9 proteins and co-inject this guide RNA protein mixture into embryos because we do find that Cas9 protein injections can not only increase efficiencies, but they also reduce toxicities that might be caused by mRNA injection. So using these platform, so our current targeting efficiency, for 70% of the genes we targeted, we can reach mutation efficiencies between 90 to 100%. And for 90% of the genes, we can reach above 50% mutation efficiency. And we also found that multiplex gene targeting is very efficient. We have tried to mix guide RNAs up to 10 different guide RNAs in the injections, and found that they don't really reduce the guide RNA efficiency. And this is especially important in zebrafish, as I mentioned previously, that there are sometimes multiple orthologs of a human gene. For example, in this case, Shanks 3A and 3B, so you may want to knock out both orthologs at the same time. And again, this is the fragment, fluorescent fragment, PCR fragment length analysis. You can see that in the co-injected embryos, the original wild type alleles is completely gone. So in addition to just knock out the genes, since we want to use zebrafish for study of human diseases, and sometimes making precise mutations are important. So for this, we have taken this approach using single-strand oligonucleotide, DNA nucleotides, SSODN, as the donor. And using this approach, we can introduce small, in this case, 40 base pair insertions into the target site. And it is important to know, if you ever want to use this approach in fish, there's some differences in terms of fish model versus the human cell models. In human cells, you usually use much longer homologous arms, but in fish, it is found that the short homologous arms are actually more effective. And so normally we use around 25 nucleotide of homologous arms, homology arms, for this approach. In addition, using the SSODN, you can also introduce single base pair substitutions, like, for example, you're studying human mutations or SNPs. So this is a great strategy. However, there's also another differences is that compare fish model to human cells, is that sometimes in the integrated alleles, you actually do find erroneous sequences, mutated sequences near the edge of the oligos. This is not seen often in human cells, I don't think, but this is quite frequently seen in fish, so you may need to screen a few more fish in order to find the one that has precise mutation. And I think that these results may suggest that these oligos are actually integrated into the zebrafish genome by this microhomology-mediated end joining instead of more traditional homologous recombination mechanisms. And this is possibly because during early zebrafish embryos, cell divisions happen so quickly, it is likely that the HR DNA repair mechanisms are not very active in that very early stage. And in contrast, the NHEJ and MMEJ may be the dominant DNA repair mechanism. So based on that, the approach to knocking more bigger fragments into zebrafish genome, we use this microhomology-mediated DNA, hopefully to engage this microhomology-mediated DNA repair, so you can use the traditional DNA donor vectors, but with much shorter homology arms, usually around 40 nucleotides. And since injection of longer DNA fragments into zebrafish embryos can cause significant toxicities, so normally the strategy is that you also introduce guide RNA target sites as a bait sequence for in vivo linearization of these vectors and then expose the microhomology arms, and that would cause the integration of the longer DNA sequences that you want to incorporate into the target site. So the approach using homologous recombination to modify precise addition of genomes can also be achieved in zebrafish using the traditional donor vectors containing about 1 kb homologous arms. In this report, in this study reported by David Grunwald's lab, they show that you can rescue or you can rescue a albino phenotype and by using a donor DNA that can fix the DNA mutations on the pigmentation genes. And in this study, they use both the TALENs and CRISPRs and show that you can achieve the homologous recombination using traditional ways. So this will allow you to do more complicated genome engineering. And conditional CRISPR knockouts are also can also be achieved in zebrafish model by making transgenic zebrafish lines that express either tissue-specific Cas9 under the control of tissue-specific promoter or inducible promoter, and then also transgenic fish with guide RNAs under the control of the U6 promoter. So in general, we do find that most of the time, the nucleases that work in culture cells also work in the fish. However, you have to be careful because sometimes there is some differences, things that possibly due to the fish are raised in a temperature 28 degrees in contrast to 37 degrees of human cells. So we do sometimes see a little bit differences in the nucleases efficiency. But in this study, I think you also are probably familiar with. In this study, the Keystrong's lab, they use bacterial selection methods to identify engineered Cas9 that exhibit altered PAM requirements. And we also tested this VQR. As you can see, this VQR mutation caught in the Cas9 can induce the binding of this protein to the NGA target site, whereas the wild-type Cas9 do not recognize the NGA PAM sequences. So we found that in fish also that the VQR Cas9 variants can also induce the mutations in the NGA target sites, as it has been found in human cells and as well as in bacteria. However, we do see very big, strong preference on the sites that contain NGAG PAM, in contrast to the NGAT and NGAA or even NGAC sites, which almost doesn't work. And I think the same preferences have also can also be seen in human cells. However, it's much stronger in the fish. So I think that CRISPR technology, applying CRISPR technology in zebrafish certainly has greatly expanded the utility of these models. I think the most interesting applications are that we can do precise mutations now to study the effect, the roles of the human mutations. But another approach is this rapid functional genomic screening approach. So we have been involved in this project in the last 3 years called Illuminating the Druggable Genome. And this is sponsored by an NIH Common Fund program. And in this project, we collaborate with David Coquelle, who is now in UCSF, and Randy, again. And I'd just like to tell you a little bit about how we apply this, and that's just to let you see what we are able to do now. And so this Common Fund project stems from this observation that today most of the drugs on the market, you can see the majority of the drugs target only targets proteins in one of these 4 protein classes, even though there are so many different protein classes, which are the GPCR, nuclear hormone receptor, ion channels, and kinases. And this is for sort of obvious reasons, because one is that these proteins usually play very important physiological roles and diverse functions in the cells. But also because, that's also because that these proteins that have more obviously druggable small molecule binding pockets, so they are targetable, they are druggable. So these 4 protein classes are then dubbed as the so-called the druggable genome. But however, if you look at all the genes, roughly 3,000 genes in these 4 protein families, and you found that currently only about 10% are being targeted as drugs. And then there are a good portion of some others that either can be targetable by chemical probes, or we have antibodies that people are studying them. But there is also still like more than 10% that nobody has ever studied them. There's no reagents to study them. And so I guess the idea of this program is that if we can know more about what they do, we may be able to identify more druggable targets and to be used in therapeutics. So I think the strength of Zebrafish is this high throughput phenotyping platform. So if we can conjugate this rapid and very efficient CRISPR knockout technology with any type of high throughput assay in the Zebrafish, then you will be able to do this rapid functional genomic screen. And in this case, we chose this behavioral phenotyping platform because they are very sensitive, and also because they have the CNS system has a very good representations of all those 4 protein family, protein classes. So in this phenotyping platforms, the embryos are arrayed into 96 well plates. Different stimuli can be applied to them. Either it's different wavelengths of lights or acoustic stimuli, touch and vibrations. And then they would give you readouts as here represented in their motions of the embryos. Very distinct signatures of this behavioral response to those stimuli, shown here for 10 different assays. And then Dave Coquelle's lab has developed these assays, and they have also put like 30,000 different compounds onto wild-type fish on these assays. So we have a reference database of all these signatures that coming from all those different compounds in the chemical library. So what we are doing right now is that we did this large-scale CRISPR knockdowns in zebrafish embryos. And then we can look at their, just directly in the injected fish, their morphological, whether they exhibit morphological phenotype, and then run them through this behavioral phenotyping assay. We obtain their behavioral signature if they have any phenotype. And then we can, from there, identify interesting mutants, but we can also look at their behavioral signatures compared with our reference chemical database. And potentially, from these comparisons, we may identify the relationship of genes, compounds, and different pathways through these results. So I think that's pretty much what I want to go through. I'm not going to talk about specific data. We have a couple interesting mutants we are following up. But then just to conclude, I think I've mentioned all our, it's a great, all the projects are conducted as a team effort. And I'm fortunate in the last 10 years, I have great technicians working on all these projects. And thank you for your attention. Thank you.
Everything is done by embryo injection, and this is not really a throughput technology. So, first question, are there other technologies available or currently under development to improve the throughput? And second, what is the throughput, especially if you think for your phenotypic behavioral platform you're now going to analyze?
So you're talking about throughput of the assay or the injections? Injections, right. So yeah, so I think a skillful technician can inject probably, I would say, 500 to 1,000, 500 embryos in a morning. Usually, embryo slate we want to inject at one cell stage because we want the mutation to be 100%, so we want to get them in as soon as possible. So usually we have about 3-hour window, meaning that we stage them, start them. We have this divider between male and females. Once you pull the divider, they lay. Like I mentioned, in the first 3 hours, they already gone through 10. So you basically only have 45 minutes when the embryos, the eggs hit the water to do the injections. So I think usually in the morning we can inject about 500 embryos. Certainly there are automated injectors. I have not used them personally. I don't think they are very popular. Usually, people still do manual injections. The throughput of the assays is much greater if we do compounds. Like I mentioned, if we do 10, we can collect 10,000 embryos. 10 embryos per well, that means 1,000 per plate. You can do, 10 plates in a morning. Each plate, I think it takes less than an hour to run through, maybe half an hour. For all those 10 assays, but when we do the gene knockdowns, yes, the throughput is lower. It's limited by the injections. It would be great that we can do electroporations. So I think that would be something that if we can get it to work. Mm-hmm.
You mentioned that the homology arm is much shorter.
Mm-hmm.
For the zebrafish, and I think there was mention about a different organism also require different lengths of the homology arm. I was curious, do we know why?
So I think I mentioned briefly that my speculation is that they actually recombined through different mechanisms. So if you use longer arms, they can recombine through homologous recombination, which is much more precise. But however, in the early development, during early development in zebrafish, the HR DNA repair mechanisms are not very active. So NHEJ and microhomology mediated end joining mechanisms are more active. So we can get them in that way much more efficiently. However, it's not that precise. So to use the microhomology engineering mechanism, you cannot have the homologous arm too long, actually. So that's why we use shorter arms, usually 25 to 40 nucleotides per side. But they are less precise, but they are more efficient during early stage.
So I love this fact because it's a very strong reflection of the broader fact that we're dependent on the biology of cells and organisms. So these fish, after the first division, the cells divide every 20 minutes.
Yeah.
And
so,
all they're doing is, if you'll pardon the expression, S and N. Okay, they're doing DNA synthesis and mitosis,
right.
And the homologous repair process is multi-step. It's precise, but it takes a while to get invasion and resolution of the recombination intermediates. And the fish genome just doesn't have time to wait around and for these orderly homologous repair events. And it's better for the fish if you think about, sort of evolutionarily, what sort of mechanism would you want to have to repair breaks during early embryogenesis? It's better to put chromosomes back together than to leave them fragmented. And back in I think it was in the 1980s, Jim Maller's lab in Colorado did an experiment with Xenopus embryos that have a similar early cleavage phase of their development. So what they did was they took these, I think, one-cell embryos, and they irradiated them very heavily, and then watched their development relative to unirradiated embryos. And they went through all the same developmental processes, making new cells. One of the reasons for that is that the cell cycle control mechanisms are not present.
There's no break.
Huh?
Yeah, there's no break.
They don't respond.
They don't respond to
the checkpoints aren't there. So the embryos are making a run for it. They're making a run for mid-blastula, where they can finally begin to undergo more orderly development. They ultimately get, as you said, to the point where they're swimming and then feeding. But in the early stages, they're trying to get to a point in development where they can evade predators and so forth. But during those stages, they're not paying any attention to what's happening to their genome. So in this experiment from Jim Maller's lab, these embryos are developing in parallel, whether their chromosomes have been fragmented or not. And when they get to mid-blastula, the unirradiated ones go ahead. The irradiated ones go, oh my God, I got no chromosomes, and they die. But they don't notice that until they get to after about 10 or 12 divisions. So, biology, right?
Yeah, so I think that's our observations as well. It's that the first few hours, the division is just so fast. There's no time to repair using homologous recombinations. And I think that mechanism usually is also more effective when it's in the S phase and mitosis. And that is so short in fish, yeah. So first of all, I think you are absolutely right. And I think that in the past, people have always been using morpholinos. And that's also how people use fish models to validate sort of human mutations and things like that. One thing when the CRISPR technology has now become more widespread, the first thing people start to notice is that there's a discrepancy between morpholino phenotype and when you actually get a mutant. Some of the morpholino phenotypes actually were not observed when you actually have a mutation, have a genetic mutations. So I think there's certainly a debate and discussions about how accurate or how specific the morpholinos effects are. I think there are many different possibilities. There's one study when people inject morpholinos into wild-type fish, you observe a phenotype. But when you make that genetic mutants and it does not have a phenotype, however, if you inject morpholino into the mutant fish, it also does not cause the phenotype you can see in the wild-type fish. So there is this difficulties, I think, in all modern organisms. It's the same thing. Even in the mice, I think in the early days when people started doing gene targeting, people say, oh my God, I made this mice and it has no phenotype. Even though all the studies tell you that this gene must be important in the pathway, but when you make the knockout mice, it doesn't have phenotype. I think there's an adaptation sometimes when you grow a mice or a fish that with the mutations start from early on, sometimes there could be compensations and from other systems. So there is certainly debates in terms of which model is better. So that's why I think that sometimes actually looking at the injected embryos directly for the phenotype is not a bad idea because you get very high efficiency. You don't have time for them to adapt. Once you grow up heterozygous fish and then breed them for homozygotes, sometimes you lose phenotypes you don't really know whether it's because of adaptation. There's also another mechanism I'm curious about in other systems, which I don't know, but I think that people have observed in some of the genetic mutants, the fish mutants, we found that there's still, proteins are still being made, even it's frame shift mutation. So there is this skipping mechanisms, not sure whether it's fish specific, but you also have to be cautious about that. I think there are some cases that have been reported that clearly you have frame shift mutations, but proteins are still being made. So